Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Fall Monday Seminar Series. Before introducing today's speaker, let me remind you that your questions can be added to the Q&A feature at any time during the presentation. No need to wait until the end of the presentation. And it's my pleasure today to introduce Holly Brown-Borg, who is a Chester Fritz Distinguished Professor in the Department of Biomedical Sciences at the University of North Dakota School of Medicine and Health Sciences. Her research examines the interplay between hormones, methylation, and gene expression in aging and age-related disease, with a focus on the somatotropic axis. And that is the topic on which she will present today. And with that, let me turn the screen over to Dr. Brown-Borg. Thank you.
Okay, it's my presentation is showing okay?
Yes.
Perfect. Great. Thank you so much for the invitation to present today. It's exciting to share our work with you, and it's great to be part of this seminar series. I really appreciate it. So I'm going to talk about somatotropic signaling today and take you through kind of a time course of some of the things that we've done over the years and what's led up to what we're currently working on. And so I always like to show this picture because I think it gives us, it's the public facing question that's always asked is, can any of these interventions such as hormones or antioxidants, any combinations of diet, can they take you from, pill taking, perhaps becoming a little bit slower, perhaps have some burden of comorbidities going on as you age, or can these things take you to a more active lifestyle and maybe happier aging, perhaps, or just a active senior life? So that's kind of the public facing question. But the better question in terms of research and is, can we really mimic metabolic factors in these long living mice that I'll talk about today? And can that lead to extended health? So just a quick overview, we're really interested in mechanisms that metabolism and stress resistance, how those are utilized to promote enhanced health and then subsequently delay aging. We use a couple of different growth hormone mutant mice, which I'll describe to you in a couple of minutes. And we think that growth hormone is really integrating many metabolic signals across the board that impacts aging in various aging processes. And overall, we believe that if you reduce somatotropic signaling, that allows the metabolism to be reprogrammed, and it really shifts resources away from growth and proliferation and more towards stress resistance and cytoprotection. So I hope to be able to show that to you today. And we think one of the outcomes of this metabolic reprogramming is a more stable epigenome. So just a little bit of background on hormones and aging and longevity. It's been there's been descriptions of hormones or circulating factors way back into ancient Roman and Chinese medical literature, and then off and on. And then in 1889, there was some publications by a physiologist, Dr. Brown Secord. At age 72, he started injecting himself self-administering dog and guinea pig testicular extracts, and said that it restored his vitality, his mental health, and his physical health. So testosterone was identified in the 1930s, and this was believed to be the active ingredient in his testicular extracts that had been shown to increase physical strength. And that's been, studies like that have been going on over the years. And more recently, there's been the TRT, which is a testosterone clinical trial, but they haven't been able to, and so in that clinical trial, they didn't find any benefits in terms of this testosterone administration to men for vitality or physical or cognitive function. So it really doesn't support the use of testosterone use for comorbidities. In 1965, however, Arthur Everett, who's at the University of Sydney, hypophysectomized rats. So removing this pituitary gland, he was able to show that it delayed the onset of many age-related pathologies in rats, such as tumor incidence, kidney aging, and cardiovascular aging. And then in 1996, our laboratory showed that growth hormone was impacting aging in mice, and we believe that this was probably one of the factors that Dr. Everett had discovered by removing the pituitary gland, removing that source of growth hormone that was actually causing a delay in age-related pathologies. So when one thinks about growth hormone, they think about growth primarily. You know, sometimes I think hormones, various factors are misnamed. But anyway, we know that it does stimulate the growth of muscle, cartilage, various organs and bones, but it also has metabolic effects. So it raises blood glucose levels. It's one of the counter-regulatory mechanisms for hypoglycemia, and it also induces fat breakdown. But with age, we know that the plasma levels of growth hormone decline, and that's probably biologically relevant to help reduce proliferative activity that leads to cancer. But there's also a loss of muscle mass and strength that occurs with age, an increase in fat mass, and a loss of bone mineral density, which are all, potentially impacted by the levels of growth hormone. And these are physical characteristics that are really associated with frailty. We also know that insulin sensitivity decreases with age. And I just show a couple of these pictures because, again, going back to the public facing question, you have individuals that are older adults that are exercising and trying to maintain muscle mass. And then you have women like Ernestine Shepherd, who is 85, that, is still winning bodybuilding championships, and she looks amazing. So there is evidence for growth hormone's effects on longevity in humans. So we know that a pathological amount of growth hormone accelerates aging and shortens lifespan. And these are in individuals that have either gigantism or acromegaly or develop a tumor that secretes high levels of growth hormone or growth hormone releasing hormone over time. We also know that growth hormone administration is associated with issues such as peripheral edema, arthralgias, glucose intolerance, diabetes, and cancer. And a lot of that growth hormone administration is being used in the sports world, and it's being promoted for use in older adults also. We think that the reduced growth hormone signaling is more important, and there is data that shows that it correlates with healthy aging and longevity. So we know that the secretion pattern of growth hormone has been linked to longevity. We also know that there's various forms of growth hormone deficiency that have been linked to have protective effects from cancer in particular. If one deletes the receptor, or if there's a receptor deletion in humans, there's a marker. It's been shown to be a marker of exceptional longevity, and we know that these receptor defects also confer protection from cancer and diabetes. So there's a population of individuals in Ecuador that have been studied, and they have a defect in the receptor, and they have high levels of growth hormone, but they can't respond to those high levels because of the receptor defect. These individuals don't develop cancer, and they don't develop diabetes despite having BMIs of over 30. So there's some evidence that if you reduce growth hormone signaling in humans, that you get a decrease in comorbidities and an increase in increase in longevity. So we think decreased growth hormone signaling is important and is associated with health and extended health and longevity in humans and also in mice. So this is some characteristics of growth hormone mutant mice. And I just draw your attention, there's several mutants available now, but the lifespan extension in animals, animal models is impressive in my opinion. So the Ames dwarf, which we'll talk about mostly today, increased life extension is seen for 50 to 70%, basically in males and females. The Snell dwarf, which is phenotypically identical to the Ames dwarf, there's a 42 to 50% increase. And then there's several other systems. There's growth hormone receptor knockout, which also has an increase of about 38 to 55% in lifespan extension. And those are very similar to those human populations where there's a receptor defect, so that they have high levels of growth hormone, but they're not able to respond to it. And one of the common things seen in all of these model systems where the animals live longer is that they have an increase in insulin sensitivity. So again, it just stresses the fact that growth hormone has significant effects on metabolic features that are important for aging also. So one thing, let me put my, I was trying to find my laser pointer, there it is. So one thing I'd like to point out is that in species that are a bit lower on the evolutionary ladder, such as flies and worms and yeast, they use insulin or IGF, insulin-like growth factor signaling, to do some of the same things that growth hormone does. So they use these factors to accumulate fuels, and that leads to that growth and proliferation. Whereas in mammals, so we're talking about mice and humans and things, there's a master regulator of growth hormone that drives insulin or drives insulin-like growth factor. And they have opposing functions, so opposing actions. So growth hormone causes lipolysis, as I had mentioned before, fat breakdown, but it blocks insulin sensitivity, whereas insulin-like growth factor has the opposite effect. It blocks lipolysis and it increases insulin sensitivity. So if you alter growth hormone pathway or the insulin IGF-1 pathway, it alters metabolism, it alters stress responses, and these decreased actions through these signaling pathways delays aging. So I'll just introduce you to the dwarf mouse. So some of you may be familiar already, but this is the Ames dwarf mouse. Here, the smaller mouse. These animals have a point mutation in a gene that drives differentiation of the pituitary gland. So they actually lack growth hormone, prolactin, and thyroid stimulating hormone. But it's been shown that growth hormone is the primary driver of this increase in lifespan through its metabolic effects and other effects that are unknown at this point. So again, these animals have a high insulin sensitivity throughout life, from birth to older age. They're highly stress resistant in terms of cellular defense. And this is the original work that was published showing that the Ames dwarf females are shown here in the blue line. These are survival curves versus the wild type females that are shown in the yellow. And also the males are shown in green, the dwarf males, and the wild type males are shown in red. And you can see there's just a delay in the in mortality or in death occurring in these mice. So in comparison to the wild type. So the curve, the shape of the curves is very similar. It's just pushed to the right or extended. And similarly, if you calorie restrict the Ames dwarf, they live about 12% longer than a dwarf that's on ad libitum food, and much longer than the wild type. Here, the yellow and the red are the dwarf on CR in yellow, the dwarf on ad libitum are in red. So they outlive a wild type animal that's on calorie restriction here in green versus a wild type animal that's been ad libitum fed. So that appears that there's overlapping and additional mechanisms that they're using in addition to the CR that extend lifespan. So they have longer health spans too. So we talk about lifespan a lot, but we also talk about health span. And we know from our work and from the work of many other laboratories that these animals are able to avoid disease, perhaps, or age-related disease. If you look in the kidney, the glomerular basement membrane thickness remains thin, youthful levels, in comparison to older animals and diabetes models. The immune system aging seems to be delayed. They develop cataracts much later than wild type animals. Neuromuscular function has been looked at, or dysfunction has been looked at a little bit, and these animals seem to maintain their function in terms of muscles much into older ages, and I'll show you a little bit of that data. Learning and memory loss, they maintain youthful levels of cognition in comparison to wild type animals. Cancer incidence is low. You've removed growth hormone and insulin-like growth factors, so they don't have that drive to cells to divide and proliferate. Glucose homeostasis is maintained in these animals. And so all of these things together, we believe, help them to avoid these age-related diseases. So again, this is our long living mouse here, the Ames dwarf. And we also know that the pathogenic aspect of growth hormone, if you raise growth hormone levels in mice, they live half as long. So these are growth hormone transgenics. So they overexpress growth hormone in the liver primarily. And they live to 12 months of age versus a wild type animal in this line that lives to 24 months of age. And they have lower stress resistance, and their insulin sensitivity is also decreased, but they still remain euglycemic. And they have just the opposite in terms of health span of the dwarf. So they have shorter health spans. So they have increased incidence of cancer. Their cognition is impaired early on when they're much younger. Their reproductive system aging is accelerated. They stop reproducing about 5 months of age, 5 to 6 months of age. And all the other things that I mentioned in terms of the Ames dwarf, it's just the opposite in the growth hormone transgenic animals. So it gives us these two model systems with no growth hormone and really high growth hormone to look at how aging processes proceed. So as I mentioned at the very beginning, the laboratory has been interested in stress resistance and metabolism. And we know that we've looked at a lot of these different systems. We've looked at scavenging systems in terms of antioxidant defense, heat shock proteins, detoxification, metal chelators, a variety of these different things. And they're very, very different between the animals that don't have growth hormone, the Ames mice, versus the animals that have very high levels. So these antioxidant defenses, mitochondrial function, I'll talk about glutathione and methionine metabolism a little bit, metallothionein, et cetera. These are upregulated in the Ames dwarf, and they're downregulated in the growth hormone transgenic mice. And this is just an example of a lot of the work that our laboratory did early on, as well as some other laboratories that have contributed, showing that the antioxidants in particular are upregulated in terms of message expression, the proteins, the activities of these different enzymes and things. And metallothionein, a metal chelator, it's upregulated. If you treat the Ames mice with growth hormone, it depresses the expression of these antioxidants. We also know that they're functionally significant. So these increases in cellular defense are significant functionally by other laboratories have treated in vivo the animals with paraquat or diquat, and the dwarf animals outsurvive the wild type animals. So we believe that this is a kind of that the antioxidant differences are functional. I mentioned mitochondrial function. Hydrogen peroxide production is low, but the enzyme, OXPHOS enzymes are all upregulated in these mice. PGC1 alpha and the PPARs are upregulated also. And some interesting work that came out of Anjay Barkey's lab showed that if you bring these animals to thermoneutrality, so we grow our animals at 22 degrees Celsius, plus or minus a bit. If you put them in 30 degrees Celsius, which is closer to their thermal neutral zone, they still live longer, not as long as they did. So it partially normalizes the long lifespan of the Ames mice. But in the growth hormone receptor knockout mice, it didn't shorten lifespan or insulin sensitivity, and the females actually lived 14% longer. So there's been a lot of discussion about animal husbandry and aging studies where we're growing these animals at a much lower temperature so that thermogenesis is being activated. So some individuals are looking at this, so it partially normalizes the Ames mouse lifespan extension. So this is some of the early work that we did. We were looking at glutathione metabolism, and we found that glutathione levels, both the oxidized and reduced levels of glutathione, were much higher. There are higher levels of GPX, so the glutathione peroxidase, and the GSSG again, which is the oxidized form of glutathione. They also had increased glutathione transferase activities. We looked at all the GSTs that we could by message expression and by protein expression and activity levels. And you can see that the increase across the lifespan. The blue bars are the Ames dwarf, and the red bars are the wild type animals at 3, 12, and 24 months of age. And they're the gene expression for GSTA1 significantly higher. And that's pretty much across the board with all these that are listed at the bottom. And also the activity levels of these GSTs when you add different substrates. So this is a wild type animal that's received saline. This is a dwarf animal that's received saline. So they already have an increased activity level. If you give the dwarf growth hormone, it knocks it down again. Just to reiterate that growth hormone is modulating the activities of some of these pathways. So the work that we had done with glutathione metabolism was really, really interesting. And we started talking to the Grand Forks Human Nutrition Center. I gave a seminar over there and started talking to the scientists that were there. And they said, well, you should be looking at methionine metabolism, and which hadn't occurred to me at the moment. And so we started working with Eric Euthus, and we're able to publish a few papers showing that the Ames dwarf really exhibits this atypical methionine metabolism. So in the literature, it's been suggested that when methionine is abundant, that the transsulfuration aspect of methionine metabolism is favored. And when methionine is low, when an individual is fasting, that the transmethylation is favored. But when doing flux assays with Dr. Euthus, we were able to show that both the methyl moiety and the carbon chain were lost much faster in dwarf mice, suggesting that both transmethylation and transsulfuration were increased in these animals in comparison to the wild type. So this is this busy pathway here, showing that all of the enzymes, so this represents the enzymes that metabolize methionine to S-adenosine methionine, etc. So they are all upregulated in the Ames dwarf, and it's not just message expression, but the protein and the activity levels of these enzymes are increased in addition. So I mentioned the transmethylation here is results from the glycine N-methyltransferase activities are regulated by growth hormone, and then transsulfuration is also increased. So the methionine is being forced down through the transsulfuration pathway, and there's less of it being recycled back to methionine because methionine synthase is actually decreased in these animals. If we administer growth hormone to the animals, it decreases the activities of these enzymes in the pathways. And this is a robust finding in our eyes, because when we look at the growth hormone receptor knockout, again, where you have that reduced somatotropic signaling, they also have increased levels of these enzymes in the methionine pathway. And when we take liver tissue from the growth hormone transgenic mice, we've shown that they have lower levels of the activities of these enzymes. And so going back to the GNMT, it seems that growth hormone may be regulating other methyltransferases in addition to the GNMT, because we find differences in BHMT and the DNA methyltransferase enzymes DNMT1 and DNMT3A. So this was a metabolomic study that we did several years ago with the Ames dwarf, where we started feeding Ames dwarf and wild type animals methionine restricted diets or methionine enriched diets. And we did this because there was some evidence from other laboratories showing that methionine restriction extended lifespans. We wanted to see the calorie restriction work had been done already, and we decided to look at methionine restriction because we knew that the methionine pathway was altered in these animals. And so the bar graphs that are in the stippled bars are the actual enzyme message expression for the enzymes in the pathway, while the metabolites are shown in the solid bars. And basically this is that methionine pathway again, and basically we see that there are genotype differences. So the dwarf are on the left and the wild type on the right. The genotype differences outweigh, or in the dwarf perform or outweigh, I guess, the differences in diet. So the crosses above each of the bars represent these genotype differences. So you can see for MAT1A, GNMT, the BHMT, and the AHCY, all of these enzymes are upregulated in the dwarf. So again, that atypical methionine metabolism. So the red bars are the dwarf on methionine restriction. So it's an 80% restriction. And the blue bars are the dwarf animals on the on methionine enrichment. So it's a 50% enrichment. And then the wild type is the same. So these are the yellow bars are the restricted, and these are the enriched. And so the stars represent differences in diets. We see a lot of differences in diet response to the diet in the wild type animals in comparison to the dwarf animals. So that's kind of the point that I was trying to make. So genotype effects are predominant with the dwarf, but the diet effects occur mostly in the wild type. And then this is the transsulfuration pathway again. It's the other half of the pathway showing the differences in the glutathione, both the oxidized and the reduced forms, and then the other enzymes involved in transsulfuration. And so we don't see differences in cysteine, but we do see a decrease in cystathionine. Again, this is a genotype effect, and homocysteine tends to be lower in the dwarf too. So there's lots of differences. And basically, and we've also looked at the growth hormone transgenic, and they have the opposite impact of the diets with high growth hormone present versus no growth hormone. So this is the work that I was talking about earlier. So many of the studies looking at calorie restriction were really focused on fat and protein, but the Ohrenreich Foundation, Norman Ohrenreich and his co-workers were really looking for strategies to reduce growth and extend lifespan. And so that's when they found that the restriction of a single amino acid in rats extended lifespan. So this is the original work that Norman Ohrenreich published, where they got an increase of 30% median lifespan and an increase of 12% in maximum lifespan in rats fed 80% reduction in methionine. And then this work was repeated in 2005 in female mice. So the early work was done in male rats, where Rich Miller decided to take a look at methionine restriction. And they started out with too low levels of methionine restriction, and they had animal death. But when they brought it to 0.15% methionine restriction at about 6 months, they saw an extension in lifespan in mice also. And then since that time, Leo Sun has also looked at lifespan extension, but he started the animals at 12 months of age, and also saw an increase in median lifespan in male mice that when methionine restriction was started later in life. So that's all very positive in terms of methionine restriction. So we also looked at methionine restriction, and which is the 0.16% versus that enriched level that I was showing you before. So this was lifespan studies using the Ames dwarf and its wild type, the growth hormone receptor knockout and its wild type, and the growth hormone transgenic and its wild type control. And what we were able to show is that there were no differences in median or maximum lifespan in dwarf animals fed methionine restriction or methionine enrichment, whereas in their wild type animals, you saw that extension that others had also seen in median and maximum lifespan. The growth hormone receptor knockout also didn't respond in terms of median or maximum lifespan differences on any of the diets, whereas their wild type again saw that extension with methionine restriction. And opposite to that are the growth hormone transgenics. So we actually saw a 50% increase in the lifespan of the growth hormone transgenics. So they were living, the wild types, the growth hormone transgenics that were exposed to a 50% reduction or a 50% enrichment didn't see any difference in lifespan extension, but when they were exposed to an 80% reduction in methionine, so that methionine restriction, they lived to 18 months versus 12 months. and then their wild types also responded with an increase. So we believe that an intact growth hormone signaling pathway is really necessary to see the differences in methionine levels. And this is just some characteristics. It's been suggested that growth hormone deficiency is just really a methionine restricted model system, and it's not really semantics. I don't believe that's the case because there's very significant differences. So they do show very similar differences in terms of lifespan extension. IGF-1 levels are low, adiponectin levels are higher, but lean mass is decreased in the dwarf animals that are restricted. The mTOR pathway is low, whereas AMPK is high, as well as fatty acid oxidation. You don't see that in methionine restriction. And then you have these other differences that are involved in glutathione. It's higher in the dwarf, it's lower in methionine restriction, et cetera, et cetera. So there's significant differences between growth hormone deficiency and methionine restriction. So the last part of the information that I want to tell you about is a little bit about some epigenetics that have been done and that are ongoing, and then about age-related diseases that we've turned to start looking at in these animals to determine how they're able to avoid or, or at least delay the presence of some of these diseases. So some of the information that I've already told you about, the dwarf exhibits this atypical methionine metabolism, and that growth hormones regulating many of these enzymes, including the DNA methyltransferases. We know that the flux of methionine is different and that transmethylation is increased. And then we also know from literature that DNA methylation is impacted by thyroid hormones, and the dwarf has low thyroid hormones. And they've shown that thyroid hormones reduce DNA methylation by upregulating the 1011 or the TETs and downregulating DNMTs, and we're seeing the opposite thing in the Ames dwarf. So again, we think that growth hormone is underlying that issue. So all of this information together suggested that maybe the dwarf epigenome was different than the wild type. So in a collaboration with Peter Adams, who at the time was in Glasgow, now he's at the Sanford Burnham Precipice Research Institute in La Jolla, we looked at liver DNA methylation in the dwarf and the wild type animals. And so we looked at 2-month-old and 22-month-old dwarf and wild type male mice. This is in liver tissue. And we were able to show that at 2 months of age, the dwarf tends to be more hypermethylated in yellow versus hypomethylated in the wild type. So these are genomic genotype type differentially methylated regions. And again, so at 2 months of age, the wild type is already hypomethylated compared to the dwarf. Then when you look at 22-month-old animals, so 20 months later, we still see that the same differentially methylated regions are hypomethylated in the wild type, many, many more than the Ames, and the Ames are hypermethylated. If we compare just the genotype differences, so these age-related differentially methylated regions, we see that the young dwarfs are more methylated than the old dwarfs. So that makes sense in terms of aging. Humans also become hypomethylated globally in with aging. And then the wild type young do have areas of hypermethylation that lose methylation with age over that 20 months when you look at the old animals. And you can see that the methylation differences in the wild type are far higher than the differences in methylation of the dwarf. So the hypomethylation is more predominant in the wild type animals. And so the most interesting graphs that out of many that we were able to generate from this data is just looking at the mean methylation per sample. So these are there were 4 animals per group. These are the young wild type in the light blue that start out a little over 60% methylation, whereas they drop to about 40% methylation in the wild type old. So you see this big drop here with aging. Whereas the dwarf young animals start out at over 70% methylation and drop to about 50% with over 20 months of age. So we see both of them decrease quite a bit, over that 20-month period of time, but the dwarf starts out higher and ends higher than the wild type. And we believe that serves as a buffer, perhaps, against age-associated hypomethylation. And then that study also started to create one of those epigenetic clocks, which I agree and disagree with. But anyway, these represent young controls. This represents young Ames dwarf, and it's the change in epigenetic age relative to what's expected. So the young Ames dwarf were younger than the controls at 2 months of age, and the old animals were still had an 18% reduction in epigenetic age compared to the wild type controls. So just another measure suggesting that the dwarf animals age at a slower rate than the wild type. So the last couple of minutes, I'm just going to take you through a couple of different disease pathways that we've been interested in over the past several years. And we took these animals, and there's a lot of individuals that work on Alzheimer's here in the biomedical sciences department. And so in collaboration with a postdoc that was in Colin Combs's lab, they had these different Alzheimer's disease mice models. And so we crossed the Ames dwarf mouse with the double transgenic, the APPPS1 double transgenics. And we found a lot of huge differences in pathology of the APP dwarf mice in comparison to the wild types and the dwarf mouse and the APPPS1s themselves. And so they were all back crossed onto a C57 black 6 background. So the animals showed lower IGF-1, so the expression of the dwarf gene, but they also had lower levels of the beta amyloid 1 through 42 and 1 through 40. Astrogliosis was lower, microgliosis was lower, and there was an increase in cytokines in the brain of these animals. So this just shows the differences. So the dwarf is here, the dwarf wild type is here, and the APPPS1 wild type is here in terms of A beta 1 to 42. This is the APP, the transgenics. They have high levels. When you have just the wild type, one copy of the dwarf gene, it's down a little bit, but is significantly decreased in the dwarf APPPS1 in comparison to the wild type. And same with both the soluble and insoluble forms of A beta 1 through 42. We also know that differentially expressed microRNAs that are associated with AD are that the microRNAs that are associated with AD indicate protection, perhaps the levels of those when we look at the dwarf APPPS1 cross in comparison to the wild types. And we also have information suggesting that the heat shock factor 1 activation is enhanced in the brains in older mice compared to wild type. So there seems to be some sort of protective aspects of brain aging when you cross to a Alzheimer's disease mouse, and in just animals that ain't straight Ames dwarf mice that haven't been crossed. There seems to be some mechanisms that provide protection. We've also have some ongoing studies looking at non-alcoholic fatty liver disease and hepatosteatosis or fatty liver in these animals. And the reason we looked at this is because we know that the PPAR alpha levels are elevated constitutively in Ames liver. And that if you put these animals on high fat, as Crystal Hill did when she was in Andrzej Bartke's lab years ago, they put animals on high fat for 12 weeks, and Ames dwarf animals maintained insulin sensitivity and low levels of pro-inflammatory cytokines in both plasma and the liver during that high-fat diet. So we've also tried to recapitulate that in culture by exposing hepatocytes to oleic acid versus BSA in wild type and dwarf mice, and also doing high-fat feeding studies, again, exposing animals to 10% versus 60% fat in wild type and dwarf mice. And this is just some of the preliminary data that we've put together. So I've been working with Tim Rhodes at the University of Wisconsin, sent him the data. We didn't have a lot of bioinformatics expertise at the time. That's been getting better here. But what we were able to show, this is in culture. These are hepatocytes in culture. And we're able to show that there's major differences in how the dwarf responds to high fat versus how the wild type responds to high fat. And this is just some histology that we did. These represent wild type animals on high fat. This is liver slices and stained with oil red O for the fat vesicles. And this is a wild type on standard diet, whereas this is a dwarf on high fat and a dwarf on standard diet. So they handle lipids differently. So we're doing some studies right now to look at lipid handling and lipid signaling in these animals. And so hopefully we get that published in the near future. And then the last thing I want to talk to you about is frailty. So I've been working with Ladora Thompson at Boston University, and she developed she reverse translated the Freed model of frailty into mice, and has been studying that on C57 black 6 mice. And so we're using that physical phenotype assessment in our mice to look at frailty and the development of frailty, to see if it's the onset's later, as we hope, earlier or later, or the same, and if that relates to their longevity and this age-related disease resistance. So we have ongoing studies looking at both biological and chronological aging, and I'll show you a little bit of that data in females. We have a graduate student working on skeletal muscle characteristics, because a lot of that hasn't been determined very well in the Ames dwarf. And we're also we're trying to identify some of the factors that potentially promote frailty, and then are going to look at the sex paradox and frailty, in which women, humans, and in mice, they become frail earlier, but they live longer in comparison to males. So we're taking a peek at some of that too. So here's some of that preliminary data. This is the biological age comparison, or chronological age comparison, I'm sorry. So these animals are both at 18 months of age, and we looked at grip strength in female dwarf compared to wild type. And when you look at chronological age, that the 18-month-old dwarf mice have higher grip strength, appear to be stronger than the wild type. We looked at rotorod to walking speed. We didn't see any significant differences. We've also looked at treadmill, so look at endurance to see how long they can walk on a treadmill. And it's we didn't see any differences in our early work here. And then when we looked at activity, which is another measure that the Thompson lab utilizes to determine frailty onset, the dwarf mice won't run on a wheel when you put them in there at 18 months of age. They just sit there and build nests underneath the wheels and do all sorts of other things besides running on them. Whereas the wild type, there was a huge variation in who ran and how long they ran and that type of thing. When we do a biological age comparison, we also see some differences. So we started these animals at 12 months of age for the wild type, and we started the dwarf animals at 18 months of age, because we see about a 50% difference in lifespan and maximal lifespan. And so we have been really interested in this biological age comparison. So we measured the wild type animals at 12, 15, and 18 months. And we measured the dwarf mice at 18, 21, and 24 months. And just after that 6 month assessment, we had 61% of the wild types had completed the 6-month assessment, whereas we had 80% of the dwarf mice complete the assessment. And so this is, again, all female data. This represents grip strength in the wild type females between 12, 15, and 18 months. We saw a decrease in grip strength over that time period. We didn't see any difference in grip strength between 18, 21, and 24-month-old animals. So this was pretty exciting. The rotorod, we didn't see any significant differences in either the wild type or the dwarf. Same with the treadmill, we didn't see any significant differences. And the activity, we just the activity levels appeared to go down in the wild type, but they weren't significant. But you can see the differences in the scale here from 0 to 10 kilometers per day, the wild type ran, whereas the dwarf, we had a couple of stars here running 2 kilometers a day, but most of the animals are not running at all. So that was a kind of our biological age comparison. And then finally, when we looked at frailty scores, and again, this is still preliminary data, and I just have this arrow pointing out that 6-month-old measures in the wild type animals versus the 0 time point measure in the dwarf animals these are 18 months old. So this is a chronological comparison. And the only thing that we can really say now with regard to frailty scores is that we had about between 64 and 72% of the animals that we tested between 12 and 18 months were robust and not considered frail in the wild type, whereas we had a higher percentage of non-frail robust animals in the dwarf at the older ages, actually. And pre-frail and frail, we had higher levels of pre-frail and frail animals in the wild type in comparison to the dwarf using the frailty assessment that Dr. Thompson developed. So these studies are ongoing. We're doing some lifespan studies looking at onset, et cetera, and trying to dive into some of the mechanisms that are involved. So all of this work together, we believe that growth hormone does modulate the metabolic responsiveness to nutrients and other metabolic pathways, perhaps. So in the presence of growth hormone, normal levels or high levels of growth hormone, animals have lower methionine metabolism. Their oxidative defense is also decreased in comparison to the dwarf. Stress resistance differs. Insulin resistance and cancer incidence are higher, and we think that the normal levels or high levels of growth hormone actually promotes aging. When you add methionine restriction, it does slow aging in those animals. But when you look at the dwarf animals and the growth hormone receptor knockout animals, they seem to have impaired nutrient sensing. They have higher levels of methionine metabolism, as I mentioned, the DNA methyltransferases. Their health seems to be enhanced, and they have this enhanced longevity. And we think in part it's due to a higher stress resistance and insulin sensitivity that's maintained in lower cancer incidence. And methionine restriction has no effect on these animals. So we believe growth hormone modulates metabolic responsiveness to nutrients and does impact other processes that are involved in aging and age-related disease. We think that the phenotypic characteristics of dwarf mice really represents some mechanisms of extended longevity or markers of biological aging that we can further explore and hopefully exploit for humans. We think that it modulates disease susceptibility in part through epigenetic mechanisms, which we're trying to look at further. So we're looking at histone modifications and other aspects of DNA methylation. So growth hormone shifts resources is our belief. So when you have growth hormone present, there's an increase of demand for nutrients for growth. And when there's an absence of growth hormone, we believe those resources are shifted towards defense mechanisms and protection. And that's how we end up with this extended health span and longevity in the Ames mice. And these are just to acknowledge everybody in the laboratory that has been working and is currently working towards the same goals here. Thank our collaborators that we've worked with in Boston and Madison and Glasgow. And in Central Florida, they did the work with the microRNAs, and always thank the foundations and the institutes and that have been helpful for supporting the work that we're doing. So with that, I would love to take any questions.
Thank you very much for that wonderful presentation. Very interesting. And there's lots of questions piling up, so I'll get right to those. First question is from Michael Lustgarten. And he says hi, and he asked which epigenetic clock indicated a reduced epigenetic age in the Ames dwarf mice when compared with controls. And then he also follows up and also asks if you could share your thoughts on epigenetic clocks.
So Tina Wong, who is working in Tony Eideker's lab, we had collaborated with them and they developed a clock. I think it was based on some earlier work that Tony had done in collaboration with Peter Adams. So then we had a publication in 2017. She was the first author that they developed a clock, and it was different than the Horvath clock. And then there's been a bazillion clocks developed since that time. So when they used the Ames dwarf, rapamycin, and calorie restricted mice, we showed that in that publication that all three of those animals, types of animals and treatments tended to show a decrease in epigenetic age, or younger animals using that clock. And I can't tell you the specifics of that clock and how, but it didn't use the same CPG island regions that the Horvath clock used. So my thoughts about the epigenetic clock. So looking at epigenetics, at DNA methylation, it's telling us something, but we just don't have the whole picture yet. So I think that all of those different clocks, the Grimm age and the Pheno age and the Horvath, all the Horvath clocks, they're showing something, but they're looking at these specific CPG island differences, and others have incorporated a few other parameters, but we don't have a biological marker yet really, or telling us what's really going on. So if you're more hypomethylated with aging, but I don't think it's telling us a whole lot of information yet. So they're all showing us the same thing, pretty much, using a few different areas on the genome. I'm just not convinced that until we have a biomarker that we can go in and say, oh, this is changing, that the information's interesting, but I don't think we're there. We don't have a solution yet to what's going on in terms of epigenetics.
Thank you. Joe Mason asked or states, it seems that a limitation of the Ames dwarf model to human relevance may be that it is a defect starting in utero and extending throughout early development. Is there data that blocking growth hormone signaling later in life has the same apparent benefits? Have you or others constructed an inducible model that enables you to block signaling once adulthood is reached?
Very good point. No, we know that all the time when we're looking at development. We don't know when aging starts, and that's true that these mice are impacted early on during development. We do know that their growth differences don't start until like 10 to 12 days of age. So they're exposed to growth hormone. Perhaps they're seeing some growth hormone signaling in utero through the female, and they're seeing growth hormone in the milk when they're nursing. But the animals about 10 days of age, their growth pattern changes. So they're the same, they're born at the same size as wild type mice. Everything's the same in growth and up to about 10 or 12 days of age. And then they just slow down completely in comparison to the wild type. So I don't know if that's when the growth hormone, postnatal growth hormone is starting to impact the system and they just don't have it. So they slow down in their growth pattern. So we realize that difference. We still think that we can see some metabolic differences that we may be able to target between those animals and the wild types that might be relevant for human aging. In terms of inducible models, we have not done that. Bill Sontag has introduced some inducible models that raise IGF-1 levels postnatally. I'm trying to think when they do them. They've done them at different ages, and they don't see the differences in lifespan by inducing a decrease after puberty or after 6 weeks of age or so. Anjay Barkey's lab has also added back growth hormone at different time points, and if they add it back after 6 weeks of age, they still maintain the differences. But if they add it before, at weaning from 3 weeks to 6 weeks of age, then it does bring the dwarf lifespan comes back closer to the wild type, but it's never fully recovered. So yes, there are differences. There's some human, there's one population of humans. It's a very small population of individuals that have the same exact PROP1 point mutation that the Ames dwarf has. And those individuals don't live any longer, they don't live any shorter than their relatives with normal levels of growth hormone, thyroid hormone, and prolactin. So it's just too small of a population to really look at a lot of, to do any studies on, perhaps. But I hope that answers your question.
Next, Larissa Constantino asks, how can we translate these interesting findings, these interesting results to humans?
Well, we're working on it. So we've tried to look at metabolic pathways and things. And now we're trying to look at age-related disease to see if we can pinpoint a couple of factors that are targetable. I don't know how else to say that. So that, we know the mTOR pathway is significantly decreased in these animals, but there's other things going on too, in addition to that. And that's why we started to we've looked at glutathione and methionine pathways and things where other people have shown big differences. But the big thing is like just try to mimic the metabolic profile through targeting specific factors, diet perhaps. We don't know yet. That's a good question.
Zhandan Wang, thanks you for a great presentation, and asked, did you see any sex differences on altered lipid metabolism and non-alcoholic fatty liver disease development in the Ames dwarf mice versus the wild type mice? And how about frailty scores? Any sex differences?
We are beginning to see some differences. The females seem to handle that high fat diet better than the males do. And I don't really have I don't have that data in front of me to tell you more about that because we're trying to get it ready for publication, but we do see sex differences, definitely sexual dimorphism present in terms of how they handle those lipids, and frailty too. So we didn't see differences in the males for either chronological aging comparisons or that biological aging comparison in males, but we do see the differences in females. So we know that growth hormone drives the levels of growth hormone that are released from, or not the levels, but how growth hormone is released from the pituitary gland differs in males and females. So females, even though the levels are similar in growth hormone, females have a kind of a low level continuously, whereas males have boluses that are seen by the liver. So they're seeing a different pattern of growth hormone impacting the liver. And we know that modifies a lot of the cytochrome P450 enzymes in the liver. So females seem to be a bit more protected than males do, and we believe it's because of that growth hormone release pattern that differs between the sexes, and then that impacts health and longevity, perhaps. But
thank you. Next, Chris Wiley says, hi, Holly. I love this story, full of links to several of our favorite pathways. Since you see links in both size and DNA methylation in dwarf mice, do you see any alterations in imprinted genes, which are both controlled by methylation and regulate growth, either by methylation or by gene expression?
great question, Chris. I haven't looked at it. We haven't done that, and I'm trying to think if anybody else has looked at imprinted genes like IGF2 or something. And I don't believe that's been done, but that would make a lot of sense. Yeah, that's a great question.
The next question is from Anonymous. So what do you think is needed to apply this research result to humans? Do you have any plans for study of this research in humans?
Another good question. I don't have plans personally, unless we would find something in one of these disease model systems that we're trying to evaluate the frailty or the fatty liver disease. If we could identify a couple of factors that are robustly changing between the dwarf and or robustly stand out between the dwarf and the wild type, that we could then look at, see if there's any drug targets out there already. For the fatty liver, we know that PPAR agonists are being used to inhibit fatty liver disease, or to slow fatty liver disease, or treat it anyway. And we know that there's differences in the dwarf in that particular pathway. But but we don't know yet. But I would love to work in humans, but I don't have access, and I don't know what we would target initially, except perhaps through diet.
We've reached 1 o'clock, but there's 2 more questions, so I will, if you can hang on with us, Holly, for one
Sure.
more minute, we can try to get these addressed. So the next one is another question from Larissa Constantino, and she asks, do the dwarf mice have any alteration in reproductive system?
Another good question, and something that I didn't mention. So the females are sterile because they are lacking prolactin, and mice need prolactin for luteal function, that's different from humans. And so we can take a pituitary gland from a wild type animal and from a donor animal and put it under the kidney capsule of an Ames dwarf, and it's so that it's no longer under negative control of dopamine. So prolactin then gets released in high levels, and then we can mate the females, and they can become pregnant and carry pups and lactate. Males are subfertile, so the thyroid hormone is really important for fertility also. And so we know that the males are subfertile. We use them for breeding all the time. They just don't know what to do right away. Their sex drive is a bit lower because testosterone levels are lower, but they are great breeders once they get started. So yes, there are fertility differences.
And the final question comes from Saidas, and she says, Holly, great talk. Nice to have you present at our seminar series. Have the sex differences in the frailty phenotypes been mapped for the changes in body composition, in particular the rate of lean and muscle mass change?
Good question. No, they have not. And so that's some work that Lador and I propose to do next. I don't have DEXA. I don't have any way to measure body composition here, unfortunately. And our Human Nutrition Research Center we talked about before I got started on the seminar is not, I don't think they're fully functional. We don't hear anything from them right now. But anyway, we don't have access, but they have, I believe, DEXA scanned for mice that we could use to start looking at body composition. But great question. Haven't done it. Would love to do it eventually, we hope.
Well, thank you very much again for a very interesting presentation.
Great. Thanks for the invitation. I appreciate it. And say hello to everybody.
Dr. Brown-Borg, I'm going to allow Barbara Shuckett-Hale to speak. She wanted to say a brief hello to you. Is that okay?
Wonderful. Yep. Thank you so much. I appreciate it.
Thank you. It was a great talk.
Thank you.
Can I speak?
Yep.
Hey,
Barbara?
Claudia.
Hey, Barbara.